Hello, everyone, and welcome to another edition of the Omics cast. My name is Albert Vilella, and for those of you that don't follow me on Twitter, here's my Twitter handle, Albert Vilella, all one word. And you can also contact me on LinkedIn if you want to discuss any of the science we're going to talk about today. And the slide deck of this presentation is available upon request. Today I'm going to talk about another Twitter thread related to GWAS, and in particular one by Sek Katirezan, who, for those who follow him on Twitter, is involved in cardiovascular disease. And so he tweeted about a recent publication paper they published on obesity and the links of the genetics score of obesity. And he answered a series of tweet replies to his initial tweet where people were saying, well, you know, how useful it is, how much can we learn from GWAS, where we sequence tens of thousands of individuals or genotype tens of thousands of individuals. We learn about 1 million SNPs per individual, and then we associate their BMI to some of these SNPs. And what he said is that there are several applications in drug discovery. One is to find the causal pathways and biomarkers, examine blood biomarkers and tails of distribution in people well prior to onset of disease, meaning you can say, well, if these people are healthy and they have these genotypes, these people have obesity or cardiovascular disease associated to obesity, and they have these other genotypes. Let's find where these genotypes are, which are the genes, and then understand the pathways and biomarkers. And two is to identify individuals at higher risk event, and three, to identify individuals with more larger relative benefit from a doctor giving them a prescription, right? So this is important because if you essentially the premise of precision medicine, if we can identify individuals that will benefit hugely from given medicine that's already available, then knowing that they will benefit hugely is important, right? And so Jeffrey Barrett, who used to be at the Sanger Institute and now it's the CSO of Genomics PLC in Oxford, replied to this Polygenic risk score Twitter thread, suggesting people to read chapter 5 of Luke Justin's PhD dissertation. Luke Justin did his PhD also at the Sanger and now is a group leader in Oxford as well. And then, so that's what I did. And I went to the PDF and that Jeff Barrett pointed to and read about what Luke Justins was proposing in understanding better some of these diseases like cardiovascular disease. And this is very similar, or it's related, reminiscent of what we discussed earlier in this cast about causal risk variance and the case or the differentiation between distinguishing a very small number of causal risk variants from the very large number of low frequency variants that have no effect on disease, right? So in a similar manner as we discussed previously, that height, a comparison of height between people in the Netherlands from people in Italy could be due or could be pointing simply to due to population stratification to the Netherlanders, the Dutch people having slightly different frequencies, and so some of these low frequency variants and Italians, completely unrelated to height, then this is the same. How do we distinguish the very small number of causal risk variants from the very large number of low frequency variants that have no effect on disease? And Luke Justins proposes in his PhD, he must have been a handful of years ago, one potential powerful tool is the study of multiplex or multiply affected families. Essentially a pedigree where different members have a condition, right? Multiplex families are more likely to harbor rare high penetrance causal variants than unrelated cases. And the variants are shared across multiple affected individuals, which allows us to whittle down the list of candidate variants by focusing only on those that are shared by many affected individuals. So the key here is, if all these thousands or millions of variants of low frequency could have an effect, how do you whittle down the ones that do have an effect? And the argument by Luke Justins is that multiplex families, whole genome sequencing, gives you that extra leverage that you can use. So this is a table of different ways in which you can filter all the SNPs and try and whittle down the ones that are associated to disease. And one of the filters is affected sharing, that a SNP or a variant is shared by at least 50% of sequenced affecteds in either subfamilies A plus B, subfamily C, or the entire family. We'll see some pedigrees later on to illustrate that. And so the idea is, as well as other ways of filtering here variants and try and associate them to disease, the filter ones, the affected sharing filter here gives us extra leverage, extra power to identify variants associated to a condition. So in his thesis, Luke Justins looks at Crohn's disease, inflammatory bowel disease, IBD, and most of the data relates to that. And then he shows in data for these multiplex families, whole genome sequencing, a case of positive gene found through this multiplex family whole genome sequencing study or analysis. The filtering here gives ordering by the maximum frequency affected in either subfamily or across the entire family, not two frameshift mutations ranked second in the list of candidates, which is good because it's a reassuring positive control, demonstrating that this method can prioritize mutations with relatively low penetrance. So here it is, an example of applying this methodology and finding a positive control that was already found by a more traditional methods. And you can follow me on Twitter if you want to ask me questions, please do, or on LinkedIn. This slide deck is available upon request, and we're going to be doing more of this where we try to explain the science that is happening in the world of genomics or omics in general, and make it more available to people to understand what's happening in this field, and how can we relate this to our day-to-day lives, how it's going to change our lives in the future. Thank you for listening, and we'll see you in the next episode.